Hey everyone, this is Avi Karn at Cornell University. I am currently a postdoc with in Invited Project, and today I'm going to talk about basically how one can use the Amplicon sequencing or AmpSeq data for marker-assisted selection. So, if you are not aware about the marker-assisted selection or Amplicon sequencing. So for AmpSeq, I have created a blog. I have a blog post on my website. It's avikarn.com. So you can go and click on the one of the tutorials I made, basically talking about the concept behind Amplicon sequencing and how to run your AmpSeq data on a CBSU, which is Cornell University Bioinformatics Facilities computer, to run the pipeline to generate the AmpSeq data. Just to get you, so in the website, basically I talk about, just the basic idea behind Amplicon sequencing, the flow chart, like what steps are involved, and once you get the data, what are the next steps? Like what files should you create, in what format, and how do you run the pipeline? So the pipeline is in command line in Linux. So yeah, just check it out. If you have any questions or feedback, let me know. Okay, so coming back to the objective of this video is to, once you get the data that is haplotype data from your markers, which is the AmpSeq data, what's next? What are the next steps? So the hap genome or haplotype data looks something that you see in the screen. Okay, so from the Analyze Amplicon pipeline, there are two important files, I would say, that I look for. So one is hap genome file, and another file it's a folder actually. It's a haplotype to FASTA folder. So in haplotype to FASTA folder, you have the sequences of the individual marker. So let's say a marker A has various alleles. So what are those alleles, and what's the haplotype ID? And another file is a hap genome file. Okay, so what I did here is I opened the file, which in a text editor, and then copied and pasted in Excel. It's because I like working in Excel. It's easier. So once I get the data, I transposed it. So in your first column, you see I have the samples. Okay, so the samples, you can see I have just said parent A. There are 4 replicates of parent A, parent B, and the progenies. Okay, just going from 1 to 63. So there are 63 progenies, and parent A and B, there are 4 replicates of each. In the header, there are markers. Each column is a marker. In this case, I'm showing you like 7 markers. So these are marker names, and so next, the one I'm pointing is this cell that I'm showing you is the haplotype ID. So 2/1 there. So parent A. So parent A for this marker is heterozygous for allele 1 and 2, and after colon, that's 9,5 is a read count of individual markers. Okay, so that's the basic format. If there's a missing data, then you would see something like I have highlighted here. It's ./.:0. So basically there was no data return. So progeny 1, for for this case, progeny 1, no data was returned. Okay, so to get started, so each haplotype ID is linked to a sequence. For example, this marker S15_3845185 is linked to an allele sequence. So what I do, so that marker is linked to a particular sequence or allele. So if I click if I open the haplotype to FASTA folder, you will see list of markers. So in this case, they're all the 7 markers. And then I open the haplotype, that file. So you can see there are 2 alleles. So C and T is linked. So allele C is coded with 1 and T is 2. So it's important in marker-assisted selection, so it's important that we know what's the desirable allele of these markers. So there are 2 alleles, what's desirable, okay? And that's on that basis, we are trying to capture the entire haplotype. So in this case, there is about over 2 million base pairs region that we are trying to capture, in our breeding. So we are capturing that entire haplotype. So the next thing, what I do usually is I make a meta sheet. You can see I have listed all the markers. So haplotype ID. Basically, what I'm saying is there can be multiple haplotype ID, but I would say desirable haplotype ID. Okay. So what's the sequence? So just for my notes, I say, okay, for this marker, I want this haplotype. And how do I know this marker is linked to this is the desirable haplotype? One can do validation. So what actual, there's several ways to identify the variation, that actual genetic variation that one may want to select for. So with this information, so I have listed all my desirable haplotype IDs, and then basically I'll make 2 more rows, and I copy those 2 columns, and I transpose. So I know that, okay, for this marker, I'm looking for allele 1 and 2. So before we go there, the reason, you might ask why you have multiple parent, why sample parents multiple times. So it's important to know if the samples is truly homozygous. Okay, so sometimes due to problem in sequencing, there's an issue with the heterozygous calling. I would say under calling. So if we have multiple samples replication, then we can tell, okay, if it's a truly homozygous or there's a heterozygous undercalling situation. For example, you can see here, parent B, this parent B is heterozygous, but the rest of the sample appear to be homozygous. So which one you do? So, something like that can show up. There are situations where, you will see in opposite, you will have, those things you have to keep in mind. Like if it's which one you trust for. Once again, and another thing is like you would notice, it's the same sample. So parent A has allele 1 and 2. So you can see the order of the allele doesn't matter since it's not phased. So just don't be confused with that when you're analyzing the data. So the next thing what I do is I highlight all the desirable, if the marker is has the desirable allele, I color code him. So basically what I do is I would highlight a column, the whole entire column, and then I just control find, and I would just say 1// and then I say replace, options, format, fill. I pick any color. You can pick any color you want. Okay, and you okay. So you do the same thing, you know, in. Okay, so you do the same thing for all the markers. So okay, so you see like you can see like I have highlighted using control and replace function, the Excel function, by to color code any marker. I mean, no, I won't say any marker if the marker is carrying the desirable allele. In this case, what I did was I highlighted any allele calls if it was carrying the desirable allele. So even if it was it didn't matter if it was heterozygous or homozygous. Okay, so you can see the pattern appearing. So okay, so I basically made a slight change in one of the markers here, because there was a slight problem. So now, once I have color coded the desirable alleles, you can see the patterns. You can see the patterns emerging. So you can see like the parent A carrying it's appeared to be having the entire haplotype region for the trait you are interested in. Okay, so I can tell this parent A is positive is carrying that haplotype. Okay, so what I would do next is just make another column. I just like to call it keep discard or you can make another column, say, I don't know, you can say something like allele status, and you can say if that all these markers have the desirable allele, you can say it's positive for that haplotype. So you can give it a positive, and the parent B does not have you can see this region, this marker has a different allele, and there's the same thing for this two markers. So I would give it a negative. So I would do the same thing again. Do the same thing for all the progeny too. Okay, minus, plus, plus, minus, plus, plus. Okay. Now I would say, I mean, we don't worry about the parents, but in this definitely I can tell this progeny is not carrying the haplotypes. I would say discard from, so the same thing with here. Keep. So keep. Let's discard. So you can see like what I did was I have, if the progeny was not carrying the haplotype for these set of markers, I have labeled them as negative. If it was, for example, progeny 7 was carrying the entire haplotype, the desired